will be a slightly more focused immune response. It's only against the spike protein. And it's early days, of course, in terms of the people who've received the vaccine as part of the rollout. Most of those have only received one dose, or everybody actually so far has only received one dose. And so it's probably too early to actually conduct those experiments. We need to collect sera very quickly and have a bank of sera to test from people who in the real world have received the vaccine. And it would be good to have that sera available from the clinical trials that have been conducted of these vaccines. And I think that Public Health England and other bodies are going to conduct that, but it would be good to be reassured that vaccine manufacturers are going to make those sera available for those tests.
And that needs to be done very quickly, one would assume, both now and for the reasons that Professor Horby said, for future variants. That just as we have accelerated the trials and the approvals, we need to be very agile in being able to test the new variants against the vaccines that are there. Perhaps finally, before I go to Carol Monaghan, Professor Horby, do you know whether this work is being done, or does it require any kind of approvals or releases of data or samples?
So it is being done. The virus is being grown, and as Wendy says, there are serum banks from natural infection. What does concern me slightly is that we really want to test the new virus against the serum raised in the vaccine trials. And my understanding is that those serum belong to the pharmaceutical companies that sponsored the trials, and permission is needed from them to use those samples for this purpose. I think it's a national priority to do that, and I think if there is any sense that the companies are not immediately giving permission for that to happen, then pressure should be borne on them.
Thank you. And who is that pressure from? Obviously, public opinion will be very engaged with this, but is it the MHRA? Is it the National Public Health Authorities?
I don't know, probably best to talk to the Vaccine Task Force, but it might be from government who have contracts with the vaccine manufacturers.
Thank you. We will follow that up after this meeting, and it's been very helpful to hear that from you and Professor Barclay. Let me turn to Carol Monaghan.
Thank you, Chair. I was hoping to ask Professor Horby.
I think he might be dealing with a dog-related incident, possibly involving a postman.
So I'll maybe come back to him in a minute. But so if I could turn then to Professor Ferguson. Professor Ferguson, you've hinted that there may be evidence that children are more susceptible to this particular strain. What evidence do you have for that?
So actually, what the data shows is we don't have a causal mechanism, but there is a hint if you carefully do what's called a case control study, carefully match cases of this variant with location and time matched cases of the non-variant and compare the age distribution. And bear in mind, this is mostly during the lockdown period in November. You see a statistically significant increased proportion of cases in under 15-year-olds for the variant compared with the non-variant. So beyond that, we know nothing. There could be a number of hypotheses why that might be the case. One of them is that maybe this variant allows, children are more susceptible to this variant. And as Wendy has said before, it's unusual that for previous strains of this virus, we know that children were less likely to get infected and certainly much less likely to get symptoms than adults, which is unusual for a respiratory virus in the sense that most respiratory viruses transmit most efficiently in children. So one possibility is this virus has changed in some way which doesn't particularly target children, just makes children more like adults a little bit, either in terms of symptoms or viral replication or transmission or both. But we have, again, this is very early days, we have very little direct biological, never mind experimental evidence that that's the case. At the moment, we have an observation that there is a slight shift in the age distribution, which will be consistent with any of those hypotheses. But I would emphasize, whilst it is a significant shift, it's not a huge shift. It's relatively small.
And when we're looking at this and looking at these cases, are we seeing poorer outcomes for children compared to the previous strain?
So there is a lot of work underway at the moment trying to look at outcomes, and you have to do that very carefully in what's called a case control design. PHE are undertaking that at the moment. Obviously, the outcomes we're most concerned with respect to hospitalizations. Hospitalization is a very, very rare outcome of childhood infection for this virus. So the focus initially will be on comparing overall hospitalization rates for this virus, overall mortality rates, and those data hopefully will become available in the coming few weeks. It will take longer to resolve things like, is there a variation in outcomes by age? Because we'll need much more data.
And obviously, how are we going to get that evidence? How are we going to accumulate the data? You have talked about hospital admissions. Are there other things that you're looking at?
So there are a lot of studies underway, we're working with PHE, other universities, London School are working with PHE. So a number of studies underway of coronavirus transmission. There's been a set of studies in schools. And whilst, of course, schools are shut now, there are residual samples which can be tested to see if they were variant or non-variant, to understand, for instance, is there any association between this variant and more transmission in schools. There are anecdotal reports that the last few weeks have seen more explosive school outbreaks in London and the south east of England, but they are just anecdotes at the moment. Then there are household transmission studies which are ongoing as part of the ONS national survey. And again, both retrospectively, those samples are being tested to see if they're variant or non-variant. Can we see a difference in the pattern of transmission in households? There's a hint that maybe there may be greater household transmission in data just literally gathered in the last day. But we don't know as yet whether that's related to age as yet or has any age effect. And of course, going forward, these studies will collect the data which allow us to distinguish between the variant and non-variant and see, are there differences in characteristics? So there's an enormous amount of work underway. This is very early, and it will take some weeks before we properly understand the characteristics of this virus, both in terms of the details of transmission, but also in terms of clinical outcomes.
And what would increased infectiousness in children, what would the implications of this be in terms of how we manage the virus?
So I would say just increased infectiousness overall in the population, if it went up evenly across all age groups, poses a challenge for control, as we know. And just that on its own, not if this virus was targeting children in any sense. If it was just an overall increase, it would still increase the challenge of maintaining, for instance, infection control in schools. Obviously, if there is a shift, and it's very much an if at the moment, such that children are now more like adults in their infectiousness than they were with the previous strains, that will pose greater risks of in transmission involving children mixing with each other. And potentially there are challenges there for particular types of control measures and what we want to do with schools.
Thank you. And if I could move now to just for a final question, Chair, to Professor Horby. You talked earlier about the virus mutations and the number of mutations that would have to happen in order for us to be where we are just now. Is there any evidence that the virus is mutating more rapidly in the UK than in other countries? Oh, you're muted, Professor.
Yeah, sorry, I don't have that data. Not to my knowledge, no. Lots of other countries also see virus mutations, but we do have much better genomic coverage than most other countries, so we're much more likely to pick it up. It may be that other countries have seen things like, or have experienced things like this, but they've not detected it because they're not sequencing the virus. And we've recently seen that South Africa has a very similar problem. They have a new variant with multiple mutations that seems to be spreading very quickly there as well. But slightly different from this one.
Can I just add to that? This is really Andrew Rambo and people like Eric Holtz in the Imperial Centre's work. But there is no evidence, for instance, that this new variant lineage, as it would be called, from a kind of genetic perspective, is evolving any faster than previous variants. And there's also no evidence of geographic variation in the rate of evolution of SARS-CoV-2 globally. I should also emphasize the South African variant, whilst it is also a variant with many changes, over 20 changes to the genome, some of which overlap, is not related to the UK variant at all. It evolved independently.
And so it's just unfortunate bad luck that we've ended up with a more infectious strain. Thank you, Chair.
Thank you. I think Aaron Bell wanted to come in on this particular point.
Yes, yeah, thank you. Just in follow-up to Carol's question, I mean, sort of Bayesian probability would urge us to be quite sceptical of the idea that the country with the best genomic monitoring should happen to be the country where the most dramatic mutations occurred. And there were clearly, I mean, you can see from the data, there's been a huge rise in cases and deaths in particularly southeastern Europe, countries like Czechia, Slovenia, and so on, that happened before this period. What, in your view, is the probability that we imported this variant rather than it developing in a mutation, in whether it's in a patient or whatever, in Kent in the first place?
Yeah, I think others can comment on this, but I mean, I'll kick off by saying we just don't know. I mean, you're right, there are other parts of the world that have high transmission rates where they will not be doing good genomic surveillance, and they may have had this strain. It could be a single importation into Kent, or it could have been de novo from a person in Kent. We just don't know. I mean, if Wendy or Neil want to add to that.
I mean, I can add a little bit to the what we know about the phylogeny of the virus. So you're completely right. We disproportionately sequence. I mean, of all global sequences of this virus, the UK has contributed nearly half. So we only have a handful, I think 10 in Denmark, a few in the Netherlands, and 2 imported into Australia, a handful of other detections of this virus lineage elsewhere. And so at the moment, if you look at the evolutionary tree of the virus, the phylogeny, the earliest known sample originates in the UK and in the east of England. And epidemiologically, it looks like a point source spreading out from a location, but we cannot completely rule out that it was an importation into that part of the country from elsewhere in the world versus arising there. And the only way we'll be able to look at that is, as further samples are collected, we look for viruses circulating now, which appear to have an earlier evolutionary time point. Obviously, we won't see the first virus, we have to infer this, but seem to predate the viruses we see in this variant in the UK. We haven't seen that as yet, so the hypothesis at the moment is it originated here, but we can't be sure.
Professor Barclay, did you want to add on that? You were nodding.
Well, maybe just a different way of looking at this is that there are so many unique mutations in this one single genome of this virus that the evidence is quite strong that that's sort of what we call within host accumulation, that a single person probably gave rise to at least many of those in that cluster. And just to point out that we know that there are other reports of similar viruses arising in immunocompromised patients, both here in the UK and elsewhere. There's a report of a particular patient in Cambridge hospitals with a different cluster of mutations, which doesn't seem to have given rise to a highly transmissible variant. But I think it's sort of just important to realise that this kind of situation will give rise to these clusters in this type of patient, wherever they are.
Thank you.
Thank you.
Just very quickly, relatedly, Chair, is the cluster that we saw in Wales, South Wales in particular, that emerged a little bit, perhaps even before the London and Kent cluster really took off, are they unrelated clusters as far as you're concerned?
I can talk to that. So we don't have as good as sequence coverage data in Wales, and so it's hard to tell, but there is this proxy for this variant, which is called what's called S-gene target failure. So with the PCR testing, which is the commonplace pillar 2 testing, community testing, not all, but a lot of the testing which we carry out targets 3 different genetic targets, engine, I think ORF1 and spike protein. And what we're seeing is with this variant, that whilst 2 of those targets are still positive, for PCR, one of them is negative because of that 6970 deletion that Wendy referred to. And so we do have evidence from South Wales, from recently collated data from the Lighthouse labs, that there is a reasonably high frequency of S-gene target failure cases in South Wales. And talking to the Public Health Wales authorities, I mean, they seem fairly convinced that some of the, at least part of the cause of the problems they're experiencing is because of this variant.
Thank you.
Thank you. Just before I go to Dawn Butler, then Graham Stringer, just on this question of the sequencing that we do, I think I heard you correctly, Professor Ferguson, saying that we sequence 50% of the sequencing done in the world. On this, is that
about 45%, I believe.
45%. So, Professor Horby, is it the case that if we are doing more sequencing than the rest of the world, we're going to expose more variants? And there's been something of an international reaction, including closing the ports, that seems to be on the assumption that we are somehow more infectious in this country, if I can put it in that way. But it may be the case that actually we are being penalised, as it were, for being more rigorous and more open about the sequencing. Is that a fair reflection?
Well, I think it is fair to say that, countries that have more extensive, more rigorous science and are very open and transparent, obviously do expose themselves to, important information being made available to others. But I think it's a global public good. It's in our interest that we know about these viruses so that we can control the spread better, and we can make sure our countermeasures like vaccines and drugs are better targeted. And it's a global, public benefit that we make that available. And what we should be doing is encouraging others to do that. And I was on a call with European colleagues just yesterday, and many of their conclusion was that they need to replicate what's being done in the UK in terms of their genomic surveillance coverage.
Absolutely.
And I also just make a comment about, so, I mean, the fact that Denmark is actually the country which does, the most per capita sequencing at the moment. But they have many fewer cases than us, and sometimes it's easier. But the fact they have picked up 10 cases with sequencing of this new variant in a country as small as Denmark with a relatively low infection rate would suggest, almost certainly in my view, that this virus has been introduced into the great majority, if not all, of European countries at the current time.
So it's already there, and presumably on the same basis, it's quite likely that there are other variants across the world, not just in European countries, that might also have high levels of infectiousness, but they're less likely to know about them because they've done less sequencing. Would that be?
So, I think the one of greatest concern other than ours at the moment is the South African one. They have much poorer epidemiological data, but there's certainly anecdotal reports of explosive outbreaks with that virus and very steep increases in case numbers. Also, Nigeria is showing very sharp uptick in case numbers at the moment, and we have no indication, we have no genetic data from there. So there's a benefit, as Peter said, globally to having better genetic surveillance.
Thank you. And just to follow up a question that Carol asked about the age distribution of infections, and I think Professor Ferguson, you were saying that we need more information on that. The Nervetag paper of last Friday, the 18th of December, said in its minutes that better data on the age distribution of infections with this variant will be available next week. In other words, this week. Have they been available? Have you had access to them yet? Have you seen them?
So, I should say, working very closely, all the university groups, Wendy's, myself, Edinburgh and others are working very closely as part of the incident management team with PHE. So we are in the process of the better data being referred to is really this S-gene target failure data from the PCR testing. But because there's a lot of variation from lab to lab in which labs actually test the S-gene, and depending on demand, samples get routed to different labs, it is taking some time to get a finalized data set there. So the results I talked about just now in terms of the age shift, I mean, I think it's true in England, true in Wales with the data we have available, but we are trying to refine those data sets as a matter of urgency to allow us to do much more rigorous case control studies.
Thank you. Professor Horby.
We're not in a position to do that right now, but we will be in the next week or so.
Next week or so. Professor Horby wanted to come in on that.
Yeah, it is reflected in the minutes of the meeting on the 21st that there has been some information come through on the age distribution, which shows a possible suggestion of slightly better transmission in children, but very preliminary. And I think Professor Ferguson's already spoken a bit about that.
Okay, thank you.
I think it's too early to make any strong conclusions about the age distribution.
All right. Dawn Butler, then Graham Stringer.
Chair, can I put on record my thanks to professors for coming in today, 2 days before Christmas. Can I just ask a question, first of all, on references to the old virus versus the new variant? In the beginning, when we discovered the virus in the UK, there were 2 different variants, one from Europe and I think the other one from the US. When we now talk about the new variant versus the old variant, what is the old variant? I'm not sure if which professor's best to answer this, actually.
Perhaps Professor Horby as chair of the group?
Yeah, I can start and say that it's very complicated because, virtually, even when you're infected with a virus, you're not actually infected with one pure strain, you're infected with a population of viruses. So even in one individual, when you sequence a virus, you're actually sequencing the dominant strain. And if you do another type of analysis called deep sequencing, you actually find that even within one individual, there's actually a population of viruses. So how you define, what virus A from virus B really depends on how deep you want to go into the genome. And it does get very complex in terms of naming lineages. And you're right, there's not an old virus and a new virus. There's certainly the new variant, which has got a characteristic fingerprint of these mutations. But there's a whole basket of other viruses which seem to behave the same mostly. But we've certainly had other variants, like the one that had a mutation that did seem to increase transmissibility a bit and has become widespread, not just in the UK but internationally. And that's become one of the dominant strains. But Wendy, as a virologist, is much better placed to explain this complexity than I am.
Professor Barclay?
I think you've done a very good job, Peter. So this virus, this new variant is called B1.1.7 because it tracks back to clade B of these. And so I think what you're referring to is in the very early stages, there were A's and B's, and then they go off and sort of branch out from there. So if you track all the way, this new one is originally derived from the clade B, so one of the 2 that you're referring to. But I think that's such a long time ago in the history of these viruses that it's probably better to compare this new one against what was around in sort of September and August, when it would appear that this new one acquired its cluster that is making it behave differently than the others around it.
Thank you. Dawn?
I would also add that, sorry, Dawn, if I can add, it's also difficult to compare in time frames because the level of interventions is different over time. So, the interventions in the summer are different from the first lockdown and the second lockdown, tier 1, 2, 3. And that obviously confounds the transmission of the virus, which is why the work that's being done and that Professor Ferguson has led a lot of is trying to compare the new variants with all the other viruses in the same time and place when they're under the same kind of pressures.
I'll just add something. So early, we've seen this before. We've seen new strains of the virus, new variants rise and dominate. D614G was perhaps the most important one, which has been proven to be also associated, peer-reviewed papers in reputable journals, including by Eric Volz in the centre I head, have shown that variant, which we don't know quite where it arose, but it was associated with the European outbreak first back in February and March, quickly dominated over older Asian lineages, which originated in China. and was one of the hypotheses why Europe saw a more explosive first wave of the epidemic than even, for instance, Wuhan did, where transmissibility appears to have been slightly lower.
Thank you. Dawn.
Similarly, in the summer, we saw, when infection levels were very low in England, we saw a new variant come in, probably imported from Spain or Europe, which is called A22V, which probably had less of a fitness advantage, but rapidly went to a high frequency in the population. And that's probably more of a founder effect, the fact a lot of infection was introduced, and then that change of transmission started in those infections on the background of lower levels of infection. But what we're seeing with this variant is quite different. We see, from a UK perspective, almost a point source at a time when we had lots of other virus around, but this one grew much more successfully in the population than all the other ones, which is another piece of the evidence for our concern about the transmissibility of this particular variant.
Absolutely. Thank you, Dawn.
Great, thank you. So the variant from Europe that quickly dominated, and we know that normally viruses mutate on average every 2 months. And just picking up on something that Carol said earlier, did you say that it's mutating at a faster rate or not, do you think?
I mean, I'll also let Wendy answer this. There's no evidence of any change in mutation rate. And I should say, these viruses accumulate mutations down each of the evolutionary lineages, each of the strains we can track, even faster than that. I mean, the rate of accumulating very significant changes may be slower, but they're mutating all the time. As Peter said, within a single person who's infected, you can find a whole population of viruses which are genetically slightly different from each other. So, I mean, it's just down to the error-prone nature of RNA viruses in how they copy. But maybe Wendy might like to add more.
Professor Barclay?
Well, just to reiterate, there is no evidence that this virus is mutating more quickly.
Okay, thank you.
Right. Professor Barclay, do we know yet the markers associated with long COVID?
No, do you mean genetic markers or clinical? I mean, long COVID is a consequence in some individuals of having been infected by this virus. There isn't any evidence that that's due to infection with a different variant of the virus. I think it's much more likely to be due to a difference in the host response that an individual mounts, which could be genetic or otherwise.
Great, thank you. And my last question, Professor Horby, you talked about a little bit about egging up, bigging up the new variant sort of thing. If we had cancelled Christmas in the beginning of December, would we have needed a tier 4?
It's hard. Yeah, I don't know. That's a policy question, and also would require some modelling assumptions. Because the assumption then is that cancelling Christmas would have had an effect on transmission that was occurring in October, November, which is when this variant arose. And so I think what would have mattered was what was being done at that time, and what we saw was that this virus managed to very successfully expand both in numbers and in space during the national lockdown, which I think speaks to the fact that it is a very efficient virus, unfortunately.
Actually, one more question. Did you in any way panic when you thought that 3 households were going to be mixing over Christmas? Did you hold your head in your hands and think, oh no, this is really a bad thing to do? What were your thoughts? Can each of the professors answer, please?
Briefly, perhaps Professor Horby first as the chair.
I think it wasn't that it's not the 3 households. I think it's it's the whole package of measures that matters. It's not just whether 3 household mix. It's also whether you've isolated before that, whether you've taken precautions before that, how intense your measures are, and whether you have, before you mixed, been careful with the social distancing, hand hygiene, face masks, etc.
Okay. Briefly, Professor Barclay and Ferguson.
Yeah, I mean, only to agree with Peter. I mean, the idea of 3 in itself is not the problem, but whether or not it was 3 on one day and then a different 2 on the next is the issue, and whether or not people were really understanding the logic behind the 3, I think is, a question. So it's about the whole before and after as well as on the day that matters.
Thank you. Professor Ferguson.
Just very briefly. So it was always a balancing act. I mean, I've said before, and it was the conclusion of all the scientific advisory bodies, that clearly any relaxation of mixing within households poses some risk. The 3-household measure, 3-household bubble, was a way of mitigating that risk, but it is an increased risk. I should say, this is on the background of the contact rates generally in the population are lower over the Christmas holiday than just before and just after. And so what we expected to see was, some increase in risk, but hopefully in a manageable way. And that comes down to a political about balancing health, protecting health against people's freedom to socialise in an important time of year.
Thank you very much.
Thank you all very much.
Thank you. Just in terms of what Professor Horby said about I think to paraphrase you, that the time that we could have acted against this new variant was in September or October when it first appeared, but obviously we didn't know about it then. This has been quite a rush, and in the last few weeks there have been a lot of information and decisive action taken. I guess, was it possible to have discovered what we've discovered in recent days and weeks much earlier, at a time that we could have nipped it in the bud, so to speak?
I think it has been detected pretty quickly, and once it was detected, I think the train of actions have been quick and quicker in the past, and in a good way. We first, PHE heard on the 8th, they told Nervetag on the 11th, we requested some actions, and then within a week we could make a decision that this was a serious concern, and then the next day measures were put in place, and then we've had, we're now here, and it's been a very quick turnaround. Whether it could have been detected prior to the 8th of December, I don't know. I think that's a question for PHE and the other surveillance networks as to whether they had access to data that would have allowed it to be detected a bit earlier.
Can I just make a quick comment on that? So most of the data which really supports the conclusion that this is more transmissible is relatively recent. It's data from November and early December. And there is despite a sequence of viruses, there is a 2 to 3 week lag time on a PCR test happening, that sample being selected and being sequenced. And so I don't think we would have had evidence. It might have been a few days earlier with the wind behind us, but it wouldn't, I think it would have been very difficult to get the same sort of level of confident conclusion that this really was associated with high transmissibility very much earlier done.
Right. And I'm just thinking about the future. We want to learn lessons on the way, and if we are going to have new variants appearing all the time, I guess the question is it possible much more quickly to discern, to detect them and discern whether they're going to be particularly dangerous, rather than needing the kind of the look back that expose the dangers of this virus?
I can start. I think there's a number of things. One is cluster investigation, where you're seeing unusual epidemiology, fast rising rates, unusual clusters, unusual size of clusters or setting of clusters, then they need to be investigated very quickly, including with genomics, to see if it's a variant of the virus. And that's how this variant was detected, through a PHE investigation of an unusual pattern of transmission in Kent. The next stage then is to really understand the biology of the virus, and I think that could be accelerated, and Professor Barclay is leading some work in that area, because what we need to do then is to very quickly grow this virus and see how it replicates in different cell lines, to then test it against drugs and vaccines, etc., to see if it has a material difference in how, if the genetic difference has a material difference in how the virus behaves, and that's really the crucial information that perhaps could be speeded up somewhat. But I think Professor Barclay could speak to that.
Okay, Professor Barclay, then I'll go to Graham and then Catherine.
Yes, so as Peter was just saying, once you detect a genetic variant using the COG UK and the surveillance system, what we perhaps have not done so much until now is have a way to interpret what the genetic changes mean. And this is a very new virus, and in general, coronaviruses are understudied as a group of viruses in comparison with some others like influenza, for example, or HIV. So there's still quite a steep learning curve to know how to interpret the genetic changes and what they will mean in terms of the phenotype, the way the virus behaves. So there is a group of virologists coming together now in the UK, and of course international work is intense in this area as well. But to try to work very closely with Public Health England and COG UK to be able to advise and interpret on those changes and put in place the biological sort of systems, really, the experimental systems and the various models that will be able to sort of supplement the real-world epidemiological data to give more confidence, really, in which ones are going to make a meaningful phenotypic change.
Thank you very much.
Can I just briefly add to that?
Sorry, yes.
I think, so the COG UK consortium has been very effective in monitoring lineages and ringing alarm bells before. I mean, in terms of D614G, as I mentioned, so this is not the first time. It's the first time it's hit, let's say, public health significance, but it's not the first time. I think, though, just on the virology side, we're learning lessons. We're learning lessons in terms of rapidly linking epidemiological data to the genomic data to be able to really pick up potential changes earlier and earlier.
Thank you very much indeed. Graham Stringer.
Can I follow up with Professor Horby and Carol's earlier questions about understanding the biology of this mutation and also determining whether it's just a strong correlation or whether it's causal? Are we testing in hospitals about the rate of infection, rate of deaths from this new variation as opposed to other variations?
Yeah, thanks. A good question. There's lots of things. What we focused on first really was whether this correlation is causal, whether we think there's a biological difference in the way that the virus is growing in the population. But there are clearly other important things. One is it associated with outbreaks in particular settings, particularly in nursing homes and in hospital, and work is ongoing looking into that. Certainly it has caused some outbreaks, but at the moment it's unclear as whether it's caused more outbreaks than other variants, but Professor Ferguson's probably got more data on that. And also, the disease severity. And at the moment, there's no indication that the disease severity is any different with this virus. But these are all things that are on the to-do list and are being done through studies that really control for all the confounding factors. So again, as I said previously, we have to look at what's happening with this virus now, with what's happening with other viruses now, because the situation is different from the past. The measures that are in place are different. The infection control measures, the testing regime is different, and also the treatment regime is different. So you can't compare a death rate now with one in March because they're lower now. So we have to compare the death rates now in people with this variant, people without, and we don't yet have that data.
I'll just add very briefly. So there are a lot of studies PhD are conducting. First of all important is this case control study Peter referred to, which controls for confounders and looks for, is there any evidence of a change in severity? And there's no evidence as yet that the power of the study at the moment is not really strong enough to draw the sort of conclusions as firmly as we would like. The second is it more likely to cause outbreaks in particular settings, care homes and hospitals, schools for that matter? There were a lot of anecdotal reports in the last few weeks of increased explosive outbreaks in all of those settings. But we don't know, as Peter said, whether that's more associated with the variant or not. And so PhD are doing quite a lot of intensive work on sampling and sequencing from those outbreaks to look at whether we see any sort of signal.
Graham?
Are there, or does that study encompass looking at transmissibility within domestic settings, whether that's increasing or not?
So we have a hint of that already, and that's from the ONS survey. You'll be aware that Office of National Statistics have an infection survey which samples a large number of people, tens of thousands of people a week, and that's a household-based design. So they recruit entire households, swab everyone in the household every week, and that allows you to track infection in households to some extent, and they're now going back and they've collected retrospectively, sequencing those viruses to determine which people were infected with the variant and the non-variant. And that's providing very powerful information because it is a truly random survey. It controls for a lot of biases. And there are hints in that data both of the increase in viral load Peter referred to, but there are early hints that yes, there may be increased household transmission of this variant. I say hints, none of this is statistically significant. It is a trend in a certain direction rather than being the numbers are low and will improve as time goes on.
Just a very, very simple question that I'd like to get into the record. In the common debate and dialogue around this issue in the media, there is, transmissibility and infectiousness are used interchangeably. Can you give us precise definitions so that there is clarity?
I'm not sure if there are completely precise definitions. Transmissibility is, though, it's the thing we quantify by this R number, the reproduction number, how many secondary cases one person infects. Infectiousness is often used on a more individual level. Is somebody infectious right now, or how infectious are they? So you can maybe think about transmissibility as being a more to the population level, on average, how well does something transmit? I could be highly infectious right now, or somebody also infected could be less infectious. Maybe Wendy wants to give her virologist view.
Professor Barclay?
I think the words are used fairly interchangeably, and perhaps thinking of individuals as being infectious, but the R is sort of being the average of all of those infectious individuals. And it's also important to just bear in mind there are many ways, which I think we did allude to earlier, in which these numbers can increase. For example, if an individual becomes infectious earlier during the course of their disease, then what we call the serial interval will reduce, and therefore the virus will spread from one person to another more quickly. And that kind of change could be underpinning what we see, as opposed to an individual shedding more virus and infecting more people at a given time. So there are several different parameters which feed into this growth of the outbreak. But I think perhaps the infectiousness is on an individual basis.
Great.
And disability.
Thank you.
The whole is better.
No, that's very helpful.
Thank you. My final question to Professor Barclay. Tony Blair has suggested if this mutation is more infectious and more people are going to get infected, that rather than waiting to give people in the queue 2 doses of the injection, more people should have 1 dose of the infection. What is your view on that, Professor Barclay?
Yeah, I think that the issue with that is that the vaccine is on the basis of giving 2 doses, and the efficacy that we are hoping for is on that basis. So to change at this point, one would hope to see a lot more analysis coming out from perhaps the vaccine manufacturing clinical trial data, if that is even available, about what the effects after 1 dose are. And then you would have to go through, a different process than we currently have done. The other thing to say is that I think this kind of decision would require some modelling to answer the question if you simply doubled the vaccine doses that we have available by going from 2 to 1 each, so you vaccinate twice as many people, what effect could that indeed have at this stage on growth, if you like, of this variant virus? And I think that's a question that modelers are looking at the moment. And you know, if that didn't give any benefit and one was vaccinating in a different regime out of license, I think that would not be ideal.
Can I paraphrase that? And are you saying that it's an interesting idea, but it's too risky at the present time without more evidence?
Yes.
Thank you.
I just add, I mean, it wouldn't be permitted. So our regulator, MHRA, is authorising vaccines on an emergency basis, given certain data provided by the trials, and that on the basis that people will receive 2 doses. And it would require an entire different regulatory submission to authorise just a single dose.
Thank you. I've read the article in the New England Journal of Medicine that I think that recommendation comes from, and it's the conclusions of that article are that the study was not designed to assess the efficacy of a single dose regimen. Nevertheless, in the interval between the first and second doses, the observed vaccine efficiency against COVID was 52%, and in the first 7 days after dose 2, it was 91%, reaching full efficacy against disease with onset at least 7 days after dose 2. So presumably this is why these things should be subjected to proper analysis, and you repose confidence in the MHRA to prescribe the right regime for these vaccines, do you? Perhaps Professor Horby is the chair.
Yes, I mean, that's right. You know, you can't assume that, you know, 1 dose is as good or half as good or whatever as good as 2 doses, because the data you have is on 2 doses. And I think that there are clearly those 2 points that Wendy said. One is we don't know if it will work or not. The other one is, will it achieve your objective? How much of the population would you need to immunise to have an effect on reducing transmission rates, which is very different from what we're currently doing, which is immunising the high risk to reduce the mortality rate.
Thank you. Catherine Fletcher wants to come in, and then Erin Bell.
Thank you, Chair. Professors, just a slight change. I'm really conscious of our responsibility to get the public to understand the science. You know, we're following the science is an oft-used phrase. And when we talk about this is much more infectious, it can get lost in that it's not quantifiable. So I was wondering if we could maybe make some benchmark analyses of the R. So I confess, when I heard that you could add 0.4 to 0.9 to the R rate, as a scientist, I kind of went, So perhaps Professor Horby, what was the R for Spanish flu, which was the last very, very, very serious global pandemic? What's the R for normal flu? And how confident are you in that 0.4 to 0.9 increase? And does that take the R for this coronavirus variant to 4? I mean, where do we get to? You know, 1 person infects 4 other people. So could you perhaps talk around that, Professor Horby, to start with?
Well, I'll start, and then I'll pass on to Neil, who's the R master. I mean, what's clear is that even during the second national lockdown, we were seeing rapid growth of infections and people ending up in hospital and stress on the healthcare system. And that's what really matters, is that even with controls in place, we're still seeing increasing case numbers and pressures on the healthcare system. And if we let that continue, we know that, you know, when the R is above 1, the case numbers just keep increasing, and that eventually we would reach a point where we would, you know, far exceed the numbers in hospital from the first wave, and we would exceed the pressures on the healthcare system in the first wave. And so therefore, you know, action is needed if you're to avoid that. But Neil is much better place to talk about.
Sorry, Professor Ferguson, wrong person.
Yeah, no, it's fine.
What's the Spanish flu R?
So the R0 for Spanish flu, we think, was about 2-ish, maybe a little under, which is much less than we think the R0. The R0, this is the, if you have no control measures in place, I get criticised for that. But for this virus, for coronavirus, when it hit Europe, was probably about 3. And so in that context, yes, a 50% increase would take it all the way up to 4.5, which is a very big increase. But this is what I wanted to emphasize, that our estimates of how much more transmissible this variant is, the new variant, compared with the pre-existing strains of coronavirus, were all derived in a context where we had a lot of social distancing in place and a lot of control measures in place. And it's not necessarily easy to extrapolate then the increase in R we saw, which I agree with Peter, the non-variant viruses, the old viruses, during the recent national lockdown in England, the SPI-M groups and multiple groups coming together were estimating an R of about 0.8 to 0.9 during that lockdown. But for this new variant, we were seeing an R of about 1.2, 1.3, maybe even a little higher. And that's why we have that 0.4, 0.5 difference, maybe even larger in some areas. So one was going up, one was going down. And that, of course, translates into about we actually think it's probably about a 50% increase in R. If you go from 0.8 to 1.2, it's also a 50% increase. But it's all measured in a very particular setting with very particular data. And whilst we're confident in it in that setting, I just want to urge caution in extrapolating wildly to what it would be if we, for instance, relaxed all controls. Okay. There's more work needed on understanding that.
But the context is, it is a big number, and it's a number that grabs your attention scientifically.
And it's a concern.
Whether it's 50% more with lockdown measures, or whether it's a whole load more with unmitigated, just let it run. Yeah. What
we know is, and this is, as Peter highlighted, the conclusion from Nervetag and then the SAGE subgroup, Nervetag and SPI-M, was it was a substantial increase, and an increase which had public health consequences.
Thank you.
Right. Just one more, Chair, if I may.
Okay.
I've been listening intently to this session. It's been incredibly powerful. One of the things that we were caught slightly with at the start of the pandemic was the levels of asymptomatic transmission. So people not knowing that they're poorly and being infectious and being able to transmit it. Is there any evidence at all that this new variant increases the period of asymptomatic transmission, or has differed? Can you asymptomatically transmit for 5 days as opposed to 3? Do you have any data on that?
I can talk to that a little bit. The answer is we don't know at the moment, but the ONS survey, which is a random survey of infection and which swabs people whether or not they have symptoms, may potentially be able to start to address some of that question. Peter.
Sorry, I was waving goodbye to my wife.
Oh, okay.
Well, at least the priorities are in the correct order. Professor Barclay.
Yes, just coming back to the biological plausibility of that sort of explanation, one of the other mutations in the 23 in this cluster is that one of the whole open reading frames, one of the whole proteins of this virus is lost, a protein called orphate. And early in the pandemic, an outbreak in Singapore reported a virus that lost orphate and reported milder or more asymptomatic or mild infections with that virus than symptomatic ones. So there is a biological plausibility to what you're suggesting, that we could have spread from people who are experiencing milder symptoms. But again, very preliminary, we don't have any biology on the new variant to back that up, only to say that, very strangely, going all the way back to 2003/4, when we had SARS-1, during its circulation in humans, right towards the end, before we finally got it under control, it also picked up an orphate deletion, which seemed to circulate rather well in comparison to what had come before. So that's another alarm bell that rang for us when we looked at CogUK at the sequence here, that there was one more mutation somewhere else, even in the genome, which may be adding up to something which we should think about.
Thank you. Very briefly, if you would, Professor, we've got a lot of other ground to cover.
Yes, just to very quickly say, whilst we have confidence about the increase in growth rate, we really don't know about the disease severity or asymptomatic transmission. It could well be the same, or it could be less, or it could be more. So we shouldn't make any assumptions.
Okay. Sorry, Catherine, you're muted, I think.
I detest Zoom with a passion. The message to the public is it does transmit more. You don't necessarily, you know, if a test is offered to you, take it. There is a possibility that you could be asymptomatic and still spreading it.
Thank you very much. Aaron Bell, and then Carol Monaghan.
Thank you, Chair, and thank all three of you for your time today and all your work on Nervetag during the epidemic. Professor Horby, if I could just quickly go through the timelines. You've already set out quite a lot of what happened in the last week or two. When was the new variant first detected, as you understand it?
Well, when it first became an issue identified by Public Health England, I understand, was the 8th of December.
Right.
When they looked at tried to understand the reasons for the increase in case numbers in Kent, and they identified this cluster of this virus variant. And then when they looked back through the data, the very earliest sample with this virus was, I think it was the 21st of September.
Right. And so some people have said, in criticising PHE, Nervetag and the government's response, that this new virus was, in quotes, spotted in September. Is that an unfair misrepresentation of what's happened?
Well, it would have been present in September. That was when the first, sequence was available. But as we've said, there's thousands of variants. So to know which particular one is a problem, requires epidemiological data.
So
yes.
Perspective. There's a 2, 3, sometimes 4 week delay between someone being swabbed and the sequence becoming available. So whilst the first sequence available is in late September, the first identified case of this virus is from late September. That sequence wasn't available. I don't know the precise date, but I suspect it wasn't available till 3 weeks later. And it takes much more than just one sequence. We need hundreds and hundreds of them showing growth of the lineage before we can conclude anything about its significance. And so the data really comes from November, which starts to show the correlation between transmission of this variant and increased transmissibility overall.
Thank you, Professor Ferguson. So just to be clear, you don't think it's fair to say it was spotted in September. It was looked at later that it was present in September. Okay, thank you. And Professor Horby as well, the Genomics Consortium has been sequencing COVID samples throughout the pandemic, but it's only been retrospectively identified, as we've just said. Do you think there was any way it could have been identified earlier, given the growing numbers in Kent? Or, because we looked at the behavioural explanations first, should we perhaps have been looking for the genomic explanations earlier?
Well, I think there's always a good case to be made for more, genomic surveillance, but there's also a case can be made for investing in other things as well. I think we've demonstrated here in the UK that that's proven to be very valuable because we've identified a variant that has public health significance, not just for us, but internationally, and we've been able to act upon it. And now, you'd probably be good to talk to people from the COG UK consortium as to ways they think that might be improved. They have a certain methodology which is very robust, which is to take almost a random sample, which allows you to avoid biases, but there may also be value in more targeted surveillance in certain groups. And we have been having a discussion about should we be doing more surveillance, for example, in immunocompromised patients in hospitals who may generate concerning variants, and that may be something that should be thought about more carefully.
Thank you. And finally, in your view, Professor Horby, has the government acted in a timely way to respond to the emerging evidence?
Yes, I think they have. We sent our first note to them raising a significant concern on the 18th, and on the 19th measures were put in place.
Thank you.
Thank you very much. Carol Monaghan.
Thank you, Chair. I'm really aware of time, so I'll try and be quick, and if I could ask the witnesses to try and be quick in response. We have so much we want to ask, but very little time with you. How quickly might this new variant spread around the UK? We know that it is present all over the UK, but we know that it's still highly concentrated in the southeast. So how quickly could it spread further from the southeast? Whoever feels able to answer.
Yeah. So first of all, it is everywhere now. So I think it's not so much talking about spread at the moment, it's talking about how quickly it grows from the level it is in every place in the country to a higher level. And we are seeing growth of this variant. Certainly there's a cluster in I think in Cumbria and growth in the northwest now. As I said, the data isn't completely clear-cut, but we're seeing growth in South Wales. There's no reason to believe that it will behave differently in different places, and the rate of growth is quite significant. Now, when you look at overall case numbers, of course, we see the very rapid rise in London, the southeast and the east of England, and that's just because it's at a much higher level of all the viruses around. This virus is dominating. In other areas, it's still at a lower level, and the other old viruses are flat or even declining, but still everywhere we're seeing increases in this variant. So schools are now shut. We're in a near lockdown situation across the country. Contact rates are lower over Christmas. So, I mean, I expect, and I hesitate to make any sort of prediction, that we will see a flattening of the curve for the next 2 weeks. We'll see at least a slowing of growth, if not a kind of more constant case numbers over this quieter 2 weeks. The critical question will be what happens in January, and the extent to which we want to then make public health measures more uniform across the country if the new variant is everywhere.
Thanks. So, I mean, talk of restricting travel between different parts of the UK, is there any point in this at this stage in terms of containing what is, as you've said, all over the place at the moment anyway, or is continued travel from different around the country still going to be problematic for this?
To some extent, every little helps. And I think, I mean, first of all, Nervetag did not make policy recommendations. We presented the evidence, but Christmas is associated with a lot of travel around the country. And so it's an exceptional time of year. So restricting that does help you prevent the situation of suddenly increasing the frequency of the virus across the whole country. It's everywhere now, we can't stop it, but it's still beneficial to not let it jump up dramatically just because we're letting people move around.
Thank you. So travel restrictions, in your opinion, are useful at this stage?
I think associated with the Christmas season, let's say, I think the rationale going forward needs to be examined more carefully.
Thank you. Thank you, Professor Ferguson. Thank you, Chair.
Thank you very much. Aaron Bell again.
Thank you, Chair. Yes, it was just a brief question about what the implications are for the PCR tests. Obviously, the fact that the mutation has this deletion, I understand, means that some of the redundancy in the tests is no longer there. And it's actually been fortuitous that that's helped us pick up and survey this. Does that firstly lead to a risk of more false negative tests? And secondly, does it mean that we need to redesign the tests, whether it's Professor Barclay or Professor Ferguson want to answer that?
Barclay, actually, here.
Professor Barclay?
Well, it's fantastic that we have that redundancy there. And we've got, the ORF1AB and the N still picking up the vast majority of cases that are out there. I think an analysis would show that the number of cases which are picked up on the positivity from the S gene alone is not significant. I mean, so we're not missing tests based on this because we built in that redundancy. Whether or not we need to go back to having 3 tests to have the full redundancy that we had before, I think there's some analysis that could be done there, but I suspect that actually we're in a pretty good place with having 2 tests which work at a highly sensitive level to pick up these infections.
Maybe I'll just add something briefly on that most countries in Europe do not test for the spike. They just have 2 targets.
Right. Thank you. And that spike test itself, I mean, that's the modification, I think it's N501Y that we're most worried about with vaccines, but I'll leave that to the rest of my colleagues to ask about that.
So actually the dropout of positivity on the S-PCR is down to the deletion we talked about earlier, which is 69.7, yep. 2 bits missing. And the PCR itself doesn't necessarily tell you then whether or not you've got this 501Y there as well. There have been other variants that have emerged earlier during this pandemic that have the 6970 deletion and don't have this particular cluster. But analyses of sequencing show that 97% now of the S gene dropouts are this cluster.
Right.
Therefore, this is a very good surrogate marker for the presence of the cluster. And that, as you said, was fortuitous in a sense because it's helped Neil and others to trace how widespread the variant has become.
Thank you, Chair.
Thank you very much indeed. Dawn Butler.
Thank you, Chair. Can I just ask a quick question on vaccinations and the effectiveness of vaccinations and their ability to adapt to the new variant? I mean, the vaccines are developed differently by different companies. How would you be able to assess which vaccines can be adapted quickly to be effective against the new variant? Can I ask this to Professor Barclay, please?
Yeah, as you said, there are several different platforms that are being used by different manufacturers to produce vaccines. Probably the most sort of new platform are the mRNA vaccines. These are the Pfizer-BioNTech and Moderna. And that's a piece of nucleic acid which is synthesized. And one of the beauties really of this novel technology is its simplicity, and in theory, its adaptability, its agility to respond. So I think that there is some talking to be done, some work to be done, but some sort of optimism that that kind of vaccine platform will be rapidly able to insert a new sequence for the variant spikes as they emerge to respond. Now, that's not to say that the other vaccine platforms couldn't also do that. The other vaccine platforms, for example, the AstraZeneca Oxford is a recombinant virus. But again, it's it's the spike gene that gets inserted into this recombinant virus. It's a little bit more complicated than adapting the mRNA. And then some of the others are using protein expression vehicles as well. But they are all flexible systems that can receive new sequence information and adapt. But probably there is an order of speed and agility that probably puts the mRNA vaccines a little bit more agile than the others.
Brilliant, thank you. I think I see Catherine wants to come in there in a bit. I'll quickly answer. My last question is, information is so vital in people understanding, how to stay safe, how to keep families safe. Should our priority be to reduce crowds as much as possible in whatever the scenario is, whether it be at train stations or supermarkets? Should our priority, until we get, the vaccine rollout on a mass scale, should that be our priority? And sorry, and I've got just an additional question to that on antibodies. How important is it for us to test antibodies in people? Thank you.
I can start with that. I think the principles haven't changed because of this virus variant. The way it's transmitted will be the same. It'll be respiratory droplets, perhaps some aerosol transmission and contaminated surfaces. So all the measures that have been recommended are the ones that will work. They'll just need to be enforced much stronger. And you're right. The risk of transmission is from person to person, and if it's and the closer and longer it is, and the more it's indoors in a poorly ventilated environment, the higher the risk of transmission. So we need to just keep reinforcing those messages and make sure that we put in place measures to reduce that. Such as don't mix in households if unless you really have to, and especially if there's a vulnerable person in the household. Be very careful in crowded spaces, and particularly places where you'll spend a long time, like the hospitality sector, etc. And there's the simple messages of hands, face, space. All of those still apply. In fact, they become even more important with this new variant.
Hello, Ms. Dawn Butler.
Thank you very much.
And
the antibodies, sorry.
Yeah, and the antibodies. I think it depends what there's various reasons to test the antibodies. I think the first thing we want to do is test whether the antibodies from people who've been infected and vaccinated neutralise this new virus and any other new viruses that come along, because that will tell us what's the risk of reinfection in people. and what's the likelihood that both the antibody-based therapies, convalescent plasma and monoclonal antibodies, work, and that the vaccines work. And then that is the baseline then for then interpreting antibody tests that you might use to, for example, say somebody has some protection. And so if they're re-exposed, their risk of reinfection is low and they can perhaps not self-isolate. But to have that kind of certifications, you really need to understand what level of protection the antibodies are providing.
Thank you, Dawn.
Thank you.
Catherine, I think, had a quick question on this theme.
Yes, very briefly. The timeline for adapting existing virus technology, are we talking 4 weeks, 4 months, 6 months? If we could just perhaps 1 for RNA and 2 for the mRNA type and 2 for a more conventional type like the Oxford AstraZeneca. Professor Barclay?
Yeah, I mean, again, I think this needs verification from the vaccine task force, who have a much better idea and I believe are in conversation with manufacturers about such a timeline. My understanding is that it would take a matter of weeks to make an mRNA, for example, that could be tested for immunogenicity, but you're still talking perhaps months before you'd have a vaccine that could be given at scale to the public.
Thank you.
Thank you very much. Graham Stringer.
Does the latest information we have and the data that we've got, does it mean that the tier 4 restrictions are unlikely to get the R number below 1? I think that's for Professor Ferguson.
So I think in the context of the festive break and schools being closed, we have, let's say, more confidence than we would when schools were open. But we will see really what happens in the next 2 weeks, and that will be informative. If we, hopefully, as we will do, see a significant decline in case numbers, both of the variant and non-variant, it will offer us greater comfort in terms of going into the new year, that maybe tier 4 might be sufficient. If we don't, of course, we're in a more difficult situation. So the fundamental question will be, how do we, and this is not about whether this variant affects children more or not, it's just that any increase in transmissibility, any increase in contacts in the population poses a risk of transmission. The real question will be, how do we reduce the infection risk associated with schools? But I mean, we will know much more in the coming 2-3 weeks about the case trends we see there, and that will inform decision making.
Right, thank you. Professor Ferguson, I'm sure other members of the committee are grateful for your input and all the academics we've had before us over the last 8 or 9 months. I'm interested in the structural process. The government told us that you weren't going to be part of the structure. I'm pleased you are. Can you tell us what happened?
I can't comment about number 10 comments. I mean, I agreed back in May that I would step aside from SAGE. I was never asked to step aside from anything else, and I didn't offer to, and I'm here now.
Well, we're grateful for your contribution.
And I would just add that, as chair of SAGE, I decided that Professor Ferguson to stay on. As chair of Nervetag, that Professor Ferguson to stay on Nervetag, and I'm very grateful that he agreed to.
Well done.
Thank you, Graham. Aaron Bell wants to come back in.
Yes, thank you, Chair. Pursuant to what you've just said about restrictions and the R rate, Professor Ferguson, I mean, I've seen it's characterised as this new variant has moved us from a siege position to one where we're now in a race. And what do you think the implications for the vaccine rollout process should be, assuming perhaps that the MHRA approve the AstraZeneca? Should we be absolutely prioritising vaccination above all else and trying to roll it out even more quickly than we already are?
Well, I mean, knowing the people on the vaccine task force and everybody in the NHS, I mean, I think it's been a top priority anyhow. Clearly, it adds extra urgency, and everything will be done, to speed it up to the extent possible. I would just say that I mean, this is going to be an unprecedented vaccination campaign in terms of its scale and speed. Far greater than what we do every year, say for seasonal flu, which is rolled out over the course of months. But so, I mean, there are huge logistic challenges. Yeah, I mean, clearly it makes it an even more urgent priority.
Thank you. And would you agree with that assessment that we've moved from a sort of siege situation where we can hope to suppress the virus, where now we're just slowing its growth?
Well, if it turns out to be the case, and I say it's a big if, we don't know, but if it turns out to be the case that actually it turns out to be quite difficult to keep schools open and keep control of this new variant, then of course that puts us in a much more difficult situation. We don't want children to lose out from education. So yeah, it just adds to the urgency. We don't know whether that's going to be the case at the moment. And things like mass use of lateral flow testing in schools might potentially help as well. So.
Thank you.
Thank you very much indeed. Just some final follow-up questions just to clarify some points we've talked about. So we know that this new variant now, you've got a high confidence that it's more transmissible. We don't know yet definitively whether it's more serious in terms of hospitalizations and deaths. Is it possible to predict or anticipate that? Is it the case that new variants tend to be over time less serious, if I can put it that way, or would that not be a safe assessment to make, Professor Horby?
I think it's, yeah, I mean, we don't know if it's more serious and we don't know if it's less serious. There are sort of evolutionary arguments to be made that that you have an evolutionary advantage if you are causing less severe disease as a virus, because what you want actually is prolonged viral excretion from somebody who is relatively well, and that then you'll be very successful. Since most patients, even with the normal variant, don't really get that sick, it's only a small proportion that get very unwell. There probably isn't much. I don't think much water in that case for this, and I think we just have to wait and see. I think what will happen is you get these sort of compensatory mutations. So it's trying to outcompete other viruses in terms of its ability to infect cells, and whether it causes severe illness or not is to an extent dependent on where it does that. So if it replicates very efficiently in the upper respiratory tract, but not in the lower respiratory tract, then you may end up with less severe disease. But if it replicates equally well in both, then it will be the same.
We've been able to detect the presence of this new variant, partly by seeing it spread across the country and detecting it that way. What we've also seen in recent weeks is compared to the first wave, the number of patients needing to be admitted into ICU facilities has fallen. Does that not give us the grounds to make a provisional judgment that might be associated with the increasing dominance of this new virus, at least in Kent and the southeast of England?
I think not, because we've seen that improvement or decline in case fatality rates since the start of the second wave, and across the country. So in areas where this variant was not there, and it's much more likely to be due to both the sort of transmission risks, with transmission happening more in younger people through the summer as they're mixing more, and also improvements in both diagnosis and care of patients.
So that finding predated the existence of this strain. And just to follow up on Erin's question on vaccination, in terms of the race to get people vaccinated, does your work on this new strain of the virus have any implications for who should be vaccinated first? Should it be the most vulnerable or people most likely to transmit the infection most?
At the moment, the priority chosen by the Joint Committee of Vaccination and Immunisation, JCVI, is to use the vaccine to protect the most vulnerable. And for me, that seems to me the most sensible approach, and I don't see any reason to change that.
That was based on the knowledge of the virus at a certain level of infectivity. The findings of the last few days and weeks, that doesn't change your judgment as to that prioritisation.
I can comment on that a little bit. That,
of course,
if we had huge amounts of stocks all available at once, such that we were able to kind of vaccinate everybody, then it might possibly change things. But we're not in that position. We're going to be basically vaccinating people as we get stocks, and an enormous amount of work from multiple groups has looked at what's the optimal strategy in such circumstances, and basically it's always optimal to protect people most at risk, and that's not affected by the level of transmissibility. We've looked at that.
Thank you. And just in terms of the spread of the virus across the country, obviously the figures that we get are to a certain extent lagged. People get obviously contract the infection before they test positive for it. Is it your view that it is now prevalent across the country to a significant degree, or can we still rely on local restrictions to contain it?
I think this will be a moving target. It is, at least at the regional level, it's in every region, but at a very local level, there'll be probably some areas which don't have it and some areas which do. But as I said, the pattern we're seeing in the data at the moment, using the S-gene dropout data as a proxy, is that those case numbers are increasing everywhere, but they're only in some areas, they're only a small proportion of all cases. So you don't see a big uptick among all cases, but those cases of the variant are increasing quite quickly. So I think it will be a moving target. I think the government prioritised the really high-risk areas with a high frequency of this variant first, based on the data available, but we will have to revisit those restrictions and in the coming weeks.
And should that be done on the fortnightly cycle of reviews, or does it need to be done in a different way?
I think in the current context, everyone is tracking data day by day.
Yeah, I would just reinforce that it is the early stages, and we've got some early indications that we're worrying about transmissibility and warranted action. We've still got quite a lot to learn about how the virus is behaving, both itself biologically, but also in areas where there's small numbers of cases and transmission rates are lower.
And what we've established about the fact that children are perhaps no more prone, but equally prone as adults to transmit this virus, does this have implications? Is a decision required before the beginning of the new term?
First of all, can I just slightly correct?
Yes, do, please.
Said that we don't have evidence of any change in transmissibility in children. What we have is a slight shift in the age distribution. So we don't know why that is. It could just be they're slightly more likely to develop symptoms, because in the past, children were quite unlikely compared with adults to develop symptoms. So there's a hint, and you're going on with your question, and if that pans out and it looks like transmission is enhanced in children, and we won't know that immediately, then it potentially will have consequences, yes, but it's too early to be definitive about a conclusion.
Well, that's very helpful and very clear. So we can't know yet. We don't have enough information to make that decision. Are you able to say when we might be in a position to do that? Obviously, people, children, the parents, teachers in schools, the more notice they have of this, the better.
So it's something both we and multiple other groups, as well as more kind of data being collected by PHE as a matter of pace, are working on. I mean, I can't easily give a timeline, partly because also schools are now closed, so we're having to rely on data collected in the past few weeks. I mean, I'm just saying everybody is working as fast as they can, and hopefully we will know more without doubt within a couple of weeks. Whether it's sufficient to be conclusive on this issue remains to be seen.
Thank you. And you said in one of your earlier answers, you gave a look forward. You thought that because of the measures that are in place now and because of the change in patterns of movement over Christmas, that you expected certainly the increase in cases to fall off a bit. I don't want to put words in your mouth. Could you, given you are a very prominent modeller of this pandemic, give us your forward look over the next 6 weeks, 2 months, to give your best assessment based on your modelling of what we might expect in terms of infection rates, hospitalisations and deaths.
So all I can say, the last set of SPIAM group brings together multiple modelling groups, and every week we generate the R numbers, which you hear reported each week, and we also generate medium-term projections, and those projections are saying if nothing changes, what's the trajectory? And of course the trajectory right now is a rather negative, pessimistic one. Case numbers are increasing, the overall R is 1.2 or something, hiding a lot of regional variation. So my comment that I expect to see that flatten, that rate of growth at least to slow, whether it actually we see case numbers decline remains to be seen, is just based on our understanding that over the Christmas period, contact rates in the population generally tend to be lower than either side of Christmas. People are generally not in work, schools are shut, and both of those things make a difference. Exactly what we will see is almost impossible to predict, partly because of the difficulty of quantifying precisely how transmissible this new variant is. If we start to see a significant decline over the next 2 weeks in case numbers overall, and particularly case numbers of this variant, I would still not say we're in a great position, we're in a worse position than we were before, but it offers a little bit more optimism that maybe we can keep on top of this. If we see case numbers increase, which is not my expectation, but if we do see them increase, we will be in a very difficult position, and I don't particularly have any thoughts as to what we do at that point.
Okay. Thank you. I'm grateful for that. I think it's very clear that both in terms of the analysis of the data and keeping an eye on the level of infections and its patterns, I think, as you put it, this is a daily task. It probably is going to mean for you and your colleagues and the science community, as well as people in government and in the NHS, it's going to be a pretty active season ahead of us. So on that note, can I thank very warmly the witnesses for giving up their time to appear today. It may be holiday time, but it's very clear that they're not on holiday. They've got a lot of important work to do, and we wish you well and convey our thanks to your colleagues on that. And can I thank the people who make these meetings of the committee possible, the clerk Danielle Nash and the broadcasting team and all of the clerk's team, arranging these meetings at short notice and having high calibre witnesses and the ability to convey this to the public takes a lot of effort and logistics. I'm very grateful for them breaking into their time with their families. So thank you very much indeed, everyone. I wish everyone a very Merry Christmas and call this meeting to an end. Order, order.
Proceeding has ended.